Alright everyone, so today I am sick and am not able to come in and deliver the lecture for a couple of reasons. One is that I don't want to get the people I work with in lab or you guys sick. Clearly this thing that I have is contagious since it's spread rapidly through my house. And then two is I really just don't feel good. So I'm going to deliver the lecture today by video. You'll be able to watch it on Blackboard, and let's get started. So today we're going to be talking about a couple of things. One is just a little bit about cleavage in Drosophila, and then move on to gastrulation and patterning in the fly. One piece of feedback I got from a lot of students right before the last exam was that you guys wanted more detailed learning objectives, so as you studied for the exam you would know exactly which topics to cover. And so I'm going to start doing that. I'm not going to read you this long list. I'm going to just put it as a slide at the beginning of lecture. And so when you go back to the book or you're studying for the exam, you'll know exactly what to read and understand so that you're prepared for things like Blackboard quizzes and the exams in the future. And again, I'm not going to read this thing, but you guys are welcome to use this as a study tool. I hope it helps. Um, Today, I want to start with something that we sort of skipped earlier in the semester on purpose, and that's to say that the way that we classify organisms depends on how they develop, and that's pretty obvious. So we call a shark because it develops into what we have called a shark, and we call a tree because it developed into what we call a tree. But to be eukaryotic means, of course, that the cell has a nucleus within it. To be multicellular means that when cells divide by mitosis, they hang around near each other and work together to perform a larger function. And to be metazoan means to be animal. And to be animal means to undergo gastrulation. So gastrulation is this really important phenomenon that only occurs in animals. And I'll also use that term metazoan or uh, similar terms to describe animals throughout this lecture, and probably it will slip into other lectures in the future. So just be familiar with the term metazoan. So this is on page 153 to 155 in your textbook. It's just a quick refresher on animals. And these are actually really important developmental concepts that I'm kind of throwing in now because now is when we're starting to talk about gastrulation in a little more detail. I want to remind you of this, which I just told you in the last slide, and and tell you that diploblasts, which is a vocab term you should know, have two germ layers. So they just have endoderm and ectoderm. They don't have the middle layer, the mesoderm, in there. Some examples of these are jellyfish and hydra, and they live up here on the top of this taxonomy of animals. So they are kind of some of the first branches off of the taxonomy of animals. And then we begin to have the triploblast animals, so these are the flatworms down to vertebrates, us. They have the three germ layers that we've talked about. They are kind of divided into two categories. One is the protostomes, or mouth first. So when these organisms are developing, one of the first structures they form is the mouth. One of the second structures, or later structures that they form, is actually what will be the anus in the future adult organism. So these guys form the mouth first, and they live up here. These are flatworms, nematode worms, and arthropods like Drosophila melanogaster. Secondly, are this group down here at the bottom, the deuterostomes, and you guys are all deuterostomes because you're vertebrates, seen here at the bottom. So they form the mouth second, which means they form the anus first. And this distinction is really important because that, whether you form the mouth or anus first, has major impacts for your developmental outcome. And you can see the different types of protostomes and deuterostomes on this slide. So the big takeaway from this slide is I just want you to understand that diploblasts and triploplasts exist and understand the vocab terms protostome and deuterostome. This is another piece of background information I skipped at the time that we're going to discuss now. This is the 3 stages of commitment. This is cell fate commitment. So before I told you differentiation was the generation of specialized cell types throughout development. It's really the last step in a multi-step process going from an uncommitted cell to a differentiated cell. So if it's the last step, what are the other steps? Well, the process of going from completely uncommitted to a differentiated cell type is called commitment. It's the process of going from uncommitted to a committed state. And it has three stages. The first one is called specification. And the stages are actually defined by experimental techniques. So a cell is said to be specified when it can differentiate into a particular cell type in a neutral environment. And what this means is if you take a cell out of an organism and place it in a petri dish, and it has no other inductive signals or interactions with other cells, but it still forms a particular cell type, then that cell is said to be specified. The specified cells are often reversible, meaning that it has taken this developmental path. If you put it in a neutral environment, it will still take that developmental path. But if you give it other signals, you can actually reverse it back to an uncommitted state. So specification is reversible. The next one is determination. So a cell is said to be determined when it can be placed now in a non-neutral environment and still differentiate into the same type of cell. So non-neutral can be another place in the embryo. We kind of talked about this with optic lobe induction of lens cells. You can take optic lobes or lens cells and place them in different parts of the body, and in some cases you can still get induction of these two cell types. It can also be in a petri dish, a non-neutral environment. It can also be in a petri dish with inductive factors present. So give them FGF, give them Wnt, give them Sonic Hedgehog, and they still ignore those factors and go down their differentiation path. This process is essentially irreversible, meaning once a cell reaches the point of determination, it's been determined that it will take that path and it's not possible to go back. And then the very last step is differentiation, or differentiated. A cell type is said to be differentiated when it has now fully adopted the fate and developed along that path, and it's now functioning often in whatever the cell type functions in. So these are three important distinctions of commitment, and you should understand each of them. So I told you there are three steps in commitment. There are also three strategies of initiating commitment or specification, that first step. One is called autonomous specification. So a cell, quote, knows what to become without interacting with other cells. And this is often accomplished through having different components of the original zygotic cell be broken up into different cells as this zygote divides. So if you imagine a zygote as a single cell, this thing is divided 3 times. So now we have 8 cells. And each of these cells has inherited different components. So one of those things is this yellow part down here. This is actually yolk. And the yolk has certain factors in it, one of them being an mRNA called macho. And macho is required to make muscle. So down here in the yellow part in the neighboring cell, you have the muscle forming cells. One interesting fact about often in autonomous specification is if you have these 8 cells and you break them up into individuals, the individual will still form whatever structure it was going to. So here, this set of individuals, these are actually the same cell, there's just 2 of them, form endoderm in this case, and these lower cells have formed notochord and endoderm. Oh, I apologize. This is ectoderm. I said the wrong word. These have formed endoderm and also muscles. So if you separate these individual cells, because they have inherited these factors from the early cleavage divisions of the embryo, they now take these developmental paths to form these particular structures even without other cells being present. And so that's autonomous specification. It's very common in invertebrates, and I just told you about that. I should learn to pop these things up as I say them. A second way to initiate a commitment process is called conditional specification. This is a cell learns its cell fate through interaction with other cells. It's very common in invertebrates and actually a lot of other animals. This is induction. We've talked a lot about induction, so I'm not going to harp on this topic much longer, but I want to introduce you to one other thing. and that is the concept of a morphogen. These are really important. You should really understand what morphogens are by the end of this lecture. A morphogen is a signaling protein that determines different cell fates in a concentration-dependent manner. So this is an example. Here's a graph of the amount of morphogen on the y-axis from low to high, and on the x-axis is distance from the morphogen secreting source. So this yellow cell is releasing the morphogen, and that morphogen is diffusing over a distance. The cells that receive the highest amount of morphogen signal, these dark red cells, adopt one particular cell fate. But once you cross this threshold, this line is the threshold value, and it calls it threshold 1. This is the concentration of morphogen, indicated here, that switches between cell fates. So if you're just to the left of this threshold value, you're going to take the dark red cell fate. If you're just to the right of that value, you're going to take the light pink cell fate. And these are actually biologically true. That distinction, that mark, is very tight. The threshold values of morphogens are extremely sensitive. So slightly more morphogen, you take one cell fate, slightly less, you take another. And so these cells, the pink ones, get just a little bit less morphogen. And you can see it goes from semi-high to lower and lower and lower. Eventually this cell just has a couple of morphogen molecules touching it. And then eventually you cross threshold concentration 2 of morphogen, and now you take this blue cell fate. So these are the cells that receive the least amount of morphogen. And often there's another threshold later of cells that don't receive any morphogen. They can often have their own responses to not receiving the morphogen signal. And so a morphogen simply is an inductive signal. It signals to induce cell fate decisions in a concentration-dependent manner. That's a really important concept. So the third strategy of specification is initiating. The third strategy of initiating commitment is syncytial specification. So a syncytium is something I've kind of introduced to you so far. I just told you that Drosophila embryos are a syncytium. A syncytium is essentially one cell with lots of nuclei inside of it. So many nuclei, one cell. It's a bag of nuclei, is often called. This is very common in insects. So here I'm showing you an image. This is one Drosophila embryo here. Anteriors to the top, posteriors to the bottom. And this is one cell, but you see all these nuclei. And in fact, most of them look like they're in metaphase, anaphase of mitosis here. So they actually divide synchronously. And we'll talk about that in a few slides. But just think of it as a bag of nuclei. So when we talk about these conditional specification signals on the last slide, that's induction. So one cell type is inducing another cell type to do something. But in this case, these are just nuclei. They're not cells. So these processes of doing specification in these cells occurs in one cell. So because it's a separate type of specification, it has its own name, syncytial specification. But think of it as conditional specification, just within one big cell. So there are morphogen gradients within this one big cell. And I want you to also note this process is not a cell fate commitment decision yet, because this is just one cell. The whole cell is sitting there, but individual nuclei are actually receiving different signals. And we'll talk about that on a future slide. It's going to come up a lot today, actually. So the bicoid caudal gradient, we've sort of talked about bicoid and caudal before. Looks like this. So this is mRNA I'm showing you here. So gradient of bicoid in the cytoplasm, you have this is the syncytium. There's lots of dots of nuclei in it. There's very high levels of bicoid in the anterior, trailing off towards posterior. This is caudal, very high levels of caudal in the posterior, trailing off towards anterior. These gradients of bicoid and caudal activate specific genes in a concentration-dependent manner in these nuclei. So remember, it's nuclei, not cells. So this nucleus will turn on one set of genes, this one will turn on another, and this one will turn on another set of genes. So it's one cell, but the nuclei are responding differently to these gradients of these transcription factors. So this is what it looks like if you just break that apart, and I don't show you the gradients on top. You have anterior, posterior, You have high bicoid, high caudal, and in the middle a mix of both. This particular gradient results in, after these nuclei are cellularized, you now have head regions which got the highest levels of bicoid signal, lowest levels of caudal, thorax which gets intermediate levels of bicoid and very low but still there levels of caudal. The abdomen gets low bicoid, medium caudal, and the tail gets very high caudal, no bicoid. So those different concentrations of bicoid and caudal allow the head, thorax, abdomen, and tail to form in the embryo. Okay, so we're going to move now to flies. Excuse me. And we're going to talk about fertilization. So we just finished. Fertilization was the last topic before the last exam. So I just want to tell you there are some differences in fly fertilization. As you might expect, every organism is slightly different. One of those differences is that there's only one place where sperm can enter the Drosophila egg. It's called the micropyle, and it sits here right at the anterior of the egg. The micropyle is a tiny little hole through this really hard layer around the egg called the chorion layer. Think of it as like a eggshell. It's a really hard, thick structure. It's very hard to get off. When I work with Drosophila embryos and I want to do an antibody stain to look at structures within the embryo, I have to remove this chorion layer because antibodies can't get through it. And to do that, I soak them in bleach. And bleach is the only thing that will get rid of this chorion layer. It's an amazing structure. Another point about fertilization in flies is that Drosophila sperm are really, really long. They're creepy. They're super long. They're about 300 times longer than human sperm. So here's an adult Drosophila male, and this is one sperm tail. All this is one big long sperm tail. So their sperm are ridiculously long. And the micropyle that I showed you earlier on the egg and this long giant sperm tail actually prevent polyspermy because only one sperm can get into that tiny micropyle, and then once it's in, it's got this big giant tail that blocks up the tube for other sperm to enter. So it's actually a mechanism of preventing polyspermy in flies. And flies don't have, do I say this? Flies don't have cortical granules, so they don't do this exocytosis to release chemicals or proteins that prevent polyspermy. They just have this kind of analog mechanism. It's kind of a block the entry point mechanism. Drosophila has a special type of cleavage. So this is at the point of cellularizing. So fertilization has occurred and all those this bag of nuclei is sitting there, and then it wants to form a bunch of cells. This cleavage is called superficial cleavage, and this is that the yolk is located in the middle, and cleavage occurs only at the surface. So this is kind of superficial. You think of someone who's superficial, they're only surface level. Well, that's kind of what this means. So here's a bag of nuclei. You want to form a bunch of cells. Well, they form the cells on the outside of the bag of nuclei, and this is yolk, this blue stuff here. So that's a description of superficial cleavage. I want to introduce you to that now. There are lots of other types of cleavage in earlier embryos, and we'll cover them as they crop up throughout the rest of the semester. I've told you what a syncytium is on a previous slide, and here's just a reminder that it's just a big bag of nuclei. So the question I have for you is, how do you get thousands of nuclei in this single cell here? Well, it turns out that the way that you do that is you do many nuclear divisions without cytokinesis. So a cell, the nucleus goes through S phase very quickly. The chromosomes segregate, so now you have two nuclei, but there isn't a cell membrane that's formed between these nuclei, so they stay within one common cytoplasm. And it's very fast. Nuclear divisions 1, that's what these numbers are, the nuclear division number. So 1 through 10 average about 8 minutes each. So these things are just going bam, 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 firing very quickly. Here's a video of those processes. So now you'll see this is one embryo, there's metaphase, boom. So now that the nuclei have divided, still one cell. Metaphase, bam, there's one more division. And I'll just let it keep going as I talk, but it's a beautiful process, and they divide synchronously. So there are signals between the nuclei that say, all right, now it's time to divide right now, boom, and they go. And it's quite striking. I think it's a beautiful process. Um, so I want to point out some important syncytial blastoderm events. So this cell, this bag of nuclei, is called a syncytial blastoderm. It's a vocab term. So let's point out some things that happen here. At the ninth nuclear division, so we're going to start here. There are 5 nuclei that reach the posterior end of the embryo. This is the posterior end, anterior's up. And they cellularize. These 5 nuclei that get cellularized are called the pole cells. They're the future germ cells of the adult. These are the cells that will make sperm and egg so that the species can propagate. So the very first cells that form in the Drosophila embryo are the germ cells, and that tells you how important germ cells are for an organism. Without them, you can't propagate a species. So the fly wants to make sure very early that it sets aside those germ cells and they are treated specially and receive special factors so that they stay happy throughout development. So pole cell cellularization occurs at the 9th nuclear division. At the 10th nuclear division, these nuclei, which have been floating around in this yolk throughout the whole cell body, they begin to migrate to the outside. So you'll see them here, right on the surface of the egg. And you can see by the 11th division, they're all sitting around the outside. There's a few nuclei in the middle, but most of them are sitting around the outside. And at the 13th nuclear division, so right after the 13th, so the 13th nuclear division happens, and then boom, cells form. So we'll talk about that process of cellularization on the next slide. But I want to point out here a couple of things. One is that these are the pole cells at the 12th nuclear division, the 13th, and the 14th. They're sitting here kind of on the outside, away from the other cells as they form. And the pole cells remain segregated kind of throughout this early part of development. Okay, so these are 3 important steps in the syncytial blastoderm. At the 14th division, you now have what's called the cellular blastoderm. So it's no longer a syncytium, it's a bunch of cells, and it's still a blastoderm. So I want to talk about energids. These things are fantastic. I think they're wonderful. So the nuclei, all these nuclei floating around in this egg are not equivalent. And you kind of know that a little bit because I told you about the bicoid and caudal gradients causing different genes to be turned on depending on the anterior-posterior position of a nucleus. One way that happens is that each nucleus is surrounded by microtubules and actin microfilaments. So here are cells that are nuclei that are just dividing. And you'll see they're surrounded by this green-white layer of microtubules and actin microfilaments. And these things kind of act like plasma membranes in the sense that they surround the cell and are hanging out to kind of segregate this dark black cytoplasm that's around the nucleus, or nucleoplasm. The nuclear membrane isn't formed in this image, but once it does, it will have nucleoplasm and a small bit of cytoplasm around in this little structure called the energid. So this one little thing here is called an energid. There are many of them on the slide. And an energid is simply a little nucleus surrounded by cytoplasm, and it's blocked off from the other nuclei by these microtubules and microfilaments. And this is important because these little energids are actually the units that, when cells do form, the energid gets kind of surrounded by a plasma membrane, and as a result, you have the contents of that cytoplasm get wrapped up in that cell once it forms. And so energids are a way of segregating cytoplasm without a plasma membrane. Okay, we're clearly not doing clicker questions, so I'll just tell you the answer. Syncytial specification is a specialized type of conditional specification that occurs in a single cell with many nuclei. True. That's exactly what I just told you. All right. So now we're going to talk about forming cells. So you have all these nuclei. They've migrated at the 10th nuclear division towards the periphery of the cell, and now you want to do that process I described, which is cellularization, to turn those nuclei into a bunch of little cells. So one of the first things that happens is these structures called furrow canals elongate and pass by the layer of nuclei. So here's a furrow. This is the plasma membrane up top. a furrow canal begins to elongate down towards the base of the nuclei. So it's moving down. This is all regulated by microtubule elongation. So if you don't have microtubules or you interfere with their function, these furrow canals cannot form. So the furrow canal makes its way down past the nucleus, and then once it reaches that point, it begins to actually constrict at the base. I've lost my mouse. At the base, and this constriction looks a little better. So I like this image because the furrow canals look better. This is one from your textbook. So now you'll see this constriction. So this is one furrow canal, this is another. They both move down, and now you have to constrict these two pieces of membrane together to pinch off and form a single cell here. This process happens almost simultaneously. There's a slight delay between individual cellularization events, but within a couple of seconds, 6,000 cells form at the same time in the fly. It's a marvel of engineering. If you think about all the lipids and proteins and microtubules and ATP and all these processes required to make 6,000 cells at once, it's amazing these guys ever get made. It's beautiful. All right, I want to talk about the maternal to zygotic transition. This is really important. So this is the time when control of development gets handed over from the maternally deposited mRNAs and proteins to the zygotic genome. Up until this point, the mom is doing all the work. Her mRNAs and proteins have done all the patterning work that we've talked about. So I want to introduce a concept to you called maternal effect mutation. So when you think about genetics, if I have, in this case, plus is a normal gene, M is a mutant version of a gene, and I cross these two, this mother and this father, to each other, and they create an F1. The F1 is either wild type, heterozygous for the mutation, or homozygous mutant for the mutation. This is in zygotic acting genes. So this is when the genome of the F1 determines what the phenotype, either wild type or mutant phenotype, would be. Let's talk about maternal effect mutations. So now I have a mother who is heterozygous for the mutation, a father's wild type, or a mother who's heterozygous for a mutation and the father's homozygous mutant for the wild type. Either way, if you set up the Punnett square for this cross, you'll see that half of these offspring are plus plus wild type, are homozygous wild type, and the other half are mutant over plus. And so the result is that all of these flies are wild type because it's the genotype of the mom that determines whether the F1 has a phenotype. So that's illustrated here much better. Here I have a mom who's heterozygous for the mutation, a dad who's homozygous for the mutation. So half of the progeny from this cross have mutation over mutation, but their phenotype is wild type because it's the genotype of the mother that determines if the F1 has a phenotype. And so let's illustrate that here. If I have a mother who is homozygous mutant and cross her to a wild type father, all the zygotes genotypes are heterozygous for the mutation, but 100% of them have a mutant phenotype because the mom was mutant. And that's also illustrated here. So why is the mother's genotype influencing the mutant status of her F1 progeny? Well, if you think about a gene like bicoid, bicoid is deposited by the mother into the oocyte. Bicoid is required to establish the anterior-posterior patterning of the embryo. If a mother is mutant for bicoid, they can't form the anterior part of the embryo. Bicoid is required to form that structure. And so as a result, the embryo dies. They don't have a head, they don't develop properly, and they die. Even if the embryo itself was heterozygous for a bicoid mutant, if the mom was homozygous for the bicoid mutant, her progeny would not form a head. And that's because she is depositing that bicoid protein or mRNA into her oocytes. And without her having that functional gene, she can't do that deposition, and as a result, the progeny don't survive. So the concept of maternal effect is It's the genotype of the mom, here in blue and red boxes, that determines the phenotype of her children. And the genotype of the children does not matter. These genotypes here in the F1 do not matter because the mom's genotype determines the phenotype of her progeny. All right, complicated concept. You should make sure you understand that. Okay, so going back to maternal to zygotic transition, maternal, at that transition point, maternal mRNAs are actively degraded. So the zygote says, get out of here, mom. It's time for me to do my thing. Excuse me. This occurs during what's called the mid-blastula transition. The mid-blastula transition is defined as that time when those really fast, I told you nuclear divisions 1 through 10 happen really quickly, about 8 minutes each. Eventually they start slowing down and then become on the order of 75 minutes each. So when the nuclear divisions slow down, cells form right after nuclear division 13, and the zygotic genome begins transcribing its own mRNAs. Those three things indicate that the mid-blastula transition has occurred. This maternal to zygotic transition is incredibly important for patterning the dorsal ventral and anterior posterior axes. This is not the word axes. This is the plural of the word axes. Okay. So I'm giving you a fake maternal effect question. So we'll walk through this together. YFG, which by the way stands for your favorite gene, is a maternal effect gene. A female of the genotype YFG null over plus is crossed to a male of the genotype YFG null over plus. Which of the following is true about this cross? Remember, maternal effect means the genotype of the mom determines the phenotype of her progeny. So if the mom is heterozygous for this mutation, then her progeny will not have a phenotype because they have a functional copy of YFG. So the answer is D. 0% of F1 will have a mutant phenotype. This is a new question. A female of the genotype YFG null over YFG null is crossed to the male of a genotype plus over plus. So again, maternal effect means the mom's genotype matters, not the zygotes. So because she is null for this mutation, 100% of the F1 will have a mutant phenotype. All right, make sure you understand why that's true. So I told you that these maternal effect genes are really important for setting up the axes and the fly. Here I have a fish. What is that, salmon maybe? So I want you to learn these axes. This is really important. I'm going to use these words a lot, so I've already used them a lot in this lecture. So anterior, head, right? Posterior, tail. Dorsal is back, ventral is belly, and then you have the lateral sections, the left and right lateral. I'm not expecting you on this slide to learn mid-sagittal, horizontal planes, transverse cross-section, etc. Normally I can explain those to you when they come up in class. What I want you to know is anterior, posterior, dorsal, lateral, ventral. I should have said anterior, posterior, dorsal, ventral, and lateral. So the question I have for you is, how does a region of the embryo, these early parts of development, know that it's dorsal, ventral, anterior, posterior? How does it know these things? And to know that, the organism has to break its symmetry. So if you think of an individual fertilized egg, it's a little round ball. Somehow you got to turn that round ball into some sort of asymmetric structure. And so it can divide and remain symmetric for a while. And that happens in many species. They have equally potentialed cells. In another species, you say that these cells are different from these cells. So you've broken this symmetry of this relationship, and now you have an asymmetric cell type. So importantly, Drosophila breaks the symmetry of the embryo before fertilization, in the oocyte. All this happens in the ovary of the mother. So what does an ovary look like? Well, it looks like this. This is a picture I took of an ovary. So this is an actual image from a microscope of an ovary, and here's a cartoon which I think illustrates the concept a little better. So again, anterior, posterior. The ovary is composed of 16 to 20 of these structures called an ovariole, which is basically an egg production assembly line. Starting at the tip, you have a structure called the germline stem cell, which divides, and one of those cells from that division remains the germline stem cell, and the other one goes on to eventually make an egg. This is an egg down here towards the bottom. So each ovary, this is one ovary, has 16 to 20 ovarioles, and flies have 2 ovaries, just like humans. Ovaries dump their eggs into the structure called the oviduct, which we kind of talked about, mammalian oviducts, and the ampulla and isthmus in fertilization in the last chapter. So I want to zoom in on one ovariole. So this is an ovariole. I don't expect you to know all these structures. I just want to show you that the ovariole is this assembly of egg production, with left being anterior, right being posterior here. So this ovary has a lot of really important cells. I want to point out just a few. So one is that when these eggs are forming, the egg goes through 4 rounds of mitosis to produce 16 cells. So you're going to pretend that there are 16 cells in here from the germ cell. So this germline stem cell divides, and one of the daughters from that division goes through 16 more rounds of mitosis. And then eventually you have this oocyte, which is what will actually be fertilized by the sperm, and 15 of these cells called nurse cells. And nurse cells basically dump mRNA and protein and other key components into the oocyte. So they're basically nursing, literally, the oocyte. They're pumping in these maternal effect mRNAs and proteins that get dumped into the oocyte. Let's move on. So how does the anterior-posterior polarity of the oocyte get established? Well, initially the oocyte itself and the follicle cells, oops, I forgot to talk about follicle cells. So follicle cells are a layer of somatic cells, so they're not germline cells. This is a layer of somatic cells that surround the oocyte here. So these somatic cells are important for maintaining oocyte integrity during this development process. So initially the oocyte and all those follicle somatic cells surrounding the oocyte are uniform, so the symmetry is not broken. So how does the symmetry get broken? Well, one way is that this gene called gherkin mRNA becomes localized between the oocyte nucleus, which is here, and those follicle cells surrounding here. So this is gherkin protein in green. It's hanging out between the nucleus and the follicle cells. And in this location, it gets translated into gherkin protein. It turns out that gherkin is an EGF, epidermal growth factor, ligand, and the EGF receptor is called torpedo, one of them in flies. So gherkin, ligand, torpedo, receptor. So gherkin, green dots, torpedo, receptor, cartoon. Gherkin signals to these follicle cells through torpedo and induces these cells to take the terminal follicle cell fate. This is called induction. We know all about induction, right? So now the follicle cells do something special. In return, the terminal follicle cells, once they've been induced, signal back to the oocyte to activate protein kinase A in the oocyte. It's these little red dots here. We would call this reciprocal induction, right? So PKA gets activated, and PKA activation causes this gene PAR1 to localize to the posterior of the oocyte. So here's PAR1 in green. PAR1 gets localized specifically to this posterior part of the oocyte. And when it does that, it does something really beautiful. PAR1 organizes microtubules such that the plus end of the microtubules points posteriorly, so plus is posterior, and the minus end points towards the anterior. So these microtubules are polarized, such that plus is here, minus is here, and that's set up by this PAR1 localization here. So we've broken that symmetry. The oocyte and follicle cells are no longer uniform. You have terminal follicle cells, and the oocyte is polarized with this microtubule gradient. And this microtubule polarization results in transport of particular mRNAs to particular places in the oocyte. So one of those is bicoid. Oh, geez. So I'm going to go back so I didn't confuse you. So bicoid goes on a dynein motor towards the minus end of the microtubules, which ends up being this very anterior most end of the oocyte. So the oocyte here is in this tan yellow color. Another protein called Oscar associates with kinesin and goes towards the plus end of the oocyte. And here's bicoid mRNA right up here. This is the oocyte I'm outlining here, and bicoid mRNA is right here up next to the nurse cells. So again, bicoid, really important for establishing the anterior-posterior pattern of the eventual embryo. Remember, this is not an embryo. This is an egg. This is all happening inside the mom's ovary. She's doing a lot of work for her children. So that's how the initial stages of anterior-posterior patterning is set up in the oocyte. So now we're going to talk about dorsal-ventral patterning of the oocyte. So again, I have the oocyte nucleus. And the oocyte itself, as it matures, gets bigger and bigger and bigger. And that's because these nurse cells are dumping cytoplasm, RNA, proteins, all these things into the oocyte. And the oocyte itself is making some things. So this cell, the oocyte, gets bigger. And as it gets bigger, the nucleus moves from this, initially I told you it was over here up next to what will be the terminal follicle cells. Eventually it moves up to this dorsal anterior part of the oocyte. And again, Gherkin signals to these nearby follicle cells up here and instructs them to take a dorsal cell fate. Without that dorsal signal, without that Gherkin signal, these cells take their default fate, which is ventral. So these cells down here don't have a nucleus with Gherkin hanging out next to them, and so by default they take their ventral cell fate. I want to use this slide to do two things. One is to tell you I'm going to start another thing in this class, which is people complained a little bit that I would have things on slides and they weren't sure if they were supposed to know them or didn't have to know them. And so what I'm going to do is if I have a big purple box around something, I'll also try to remember to say it out loud. I'm not expecting you to memorize all the things in this box. What I do expect you to know are the things that I'm going to put here. So I'm going to summarize all this crazy business on the left-hand side of the slide. And I do want you to know the things that are here. Okay? So one thing is that Gherkin, up in the anterior dorsal part of the oocyte, inhibits ventral cell fate in those dorsal follicle cells. So Gherkin prevents ventral cell fate formation. Without Gherkin, the ventral cells take their default fate, which is ventral. Okay? Ventral follicle cells, due to that lack of Gherkin signal, adopt a ventral fate and signal to the ventral side of the oocyte. So now these ventral cells, these somatic epithelial cells, are signaling to the oocyte and telling the oocyte, we're the ventral side of the oocyte. Okay? I don't expect you to know all the steps in that signal. I just want you to know that there is a signal from the ventral follicle cells to the ventral side of the oocyte. Later, so after fertilization, remember these steps up till now have all been in the ovary. After fertilization, the dorsal protein is activated in the oocyte